Okay, the Biochemical Society and Portland Press are pleased to welcome you to this webinar, which is part of our Biochemistry Focus webinar series. So topics in the series will include different research areas in molecular biosciences, as well as practical sessions to support your career development. And each webinar will give you the opportunity to ask questions via text, and we welcome suggestions for future topics and speakers to feature in our webinar series in the future. So please see the Biochemical Society website for more details. My name is Ben. I'm a postdoc in the Department of Biochemistry at the University of Oxford. I'm also a member of the Early Career Advisory Panel with the Biochemical Society, and I'm very happy to be here chairing the seminar today. So the webinar is part of our dedicated Early Career Research Programme of webinars, and we are delighted to provide this opportunity today for Alessia, John, and Martina to share their work with the molecular biosciences community. Today's webinar is entitled Developments in Protein Structures and Genome Integrity. We'll hear from three very exciting talks from the three early career researchers who will share their current work in the field of genome stability and protein structures and mass photometry. Before I hand over to our first speaker, I'd like to mention that questions will be asked at the end of the webinar, but please do send in your questions during the talks. If you have a question, please type it into the question box as shown in the image on the screen, stating who your question is for, please, as well, and we will try to answer as many as possible at the end. So our first invited speaker today is Alessia Lepore from the University of Edinburgh. Alessia is a Marie Skłodowska-Curie Fellow at the Laboratory of Optics and Biosciences at École Polytechnique in France, and is a visiting scholar in the El Khoury Lab in the University of Edinburgh, UK. She's been a postdoctoral research associate at the Institute of Cell Biology at the University of Edinburgh. Alessia uses biophysical methods to investigate molecular processes in bacterial systems, and today Alessia will discuss her work on in vivo single molecule imaging of RecB during DNA damage. So I'll hand over to you, Alessia.
Thank you, Ben. Thank you for the introduction. You can see everything all right?
Yeah, all good.
Perfect, thank you. Okay. Thanks. I also would like to thank the Biochemical Society for having me today. So the kind of DNA damage we are interested in are DNA double-strand breaks. This kind of DNA damage involves both strands of the double helix, and is, if it's not repaired, could be lethal for the cell, and it's a huge threat to genome stability. This kind of DNA damage could be caused by endogenous sources such as collapsing the replication forks, but also exogenous agents such as ionizing radiation. And there is even an entire class of antibiotics, the quinolones, that aim to kill bacteria by making this kind of DNA damage. However, E. coli evolved a strategy to repair this kind of damage that relies on homologous recombination. In homologous recombination, the damaged copy of the chromosome is repaired used as template and intact and identical copy of the chromosome that is undamaged. The first player in this repair pathway is RecBCD, that finds the DNA blend's end and starts to process the DNA until it recognizes a specific DNA sequence called the Chi site. After the Chi site recognition, RecBCD will change biochemical activity and promote on the single-stranded DNA the loading of the RecA protein. The RecA protein will form a filament, and at this point, RecBCD will dissociate from the DNA. The RecA protein then will look for the homology copy of the DNA of the chromosome and eventually finalize the repair process. The RecA filament also triggers the SOS response. In this response are activated more than 40 genes, and also the cell division is arrested. And this is happen all in the effort to use the cell resources to repair DNA double-strand breaks. We were particularly interested in understand in live bacteria the first step of DNA double-strand break recognition and the first step of the repair. So we focus our attention on RecBCD. RecBCD is an enzymatic complex that has three subunits, B, C, and D, and has both an helicase and a nuclease activity. The RecB subunit is the one that has both the helicase and the nuclease domain, and therefore is a crucial subunit for the functionality of the complex. And for this reason, it's perfect to aim that is looking at the RecBCD dynamics of the DNA repair. So we focus on RecB, and we label each of the RecB subunit with the halo tag. Then the halo tag will covalently bind a very bright organic dye. We quantify the number of RecB at the single cell level, and we know that are low abundant. And this is allowed us to directly follow the dynamics of single RecB molecule in the cell. So to monitor the repair, we acquire we do streaming acquisition of RecB movement, and for each field of view, we also acquire the bright field and the GFP channel. Indeed, we insert in our strain an SOS fluorescent reporter also to monitor the SOS induction in our cell. So from the bright field, we find the outline of the cell, so we segment the cell in our sample, and we use the cell segmentation to perform the tracking at the single cell level. And this is also allow us to quantify the SOS signal. In this way, we obtain quantitative information on our sample. We have indeed information on RecB mobility, on the cell size, and on the SOS signal. And in this way, we are monitored two steps of the repair, the initial DNA double-strand break recognition and the later SOS induction linked to the formation of the RecA filament. So we induce different level of sublethal of DNA damage by using sublethal concentration of ciprofloxacin. This is one of the antibiotics of the quinolones family. In the plot, I'm going to show you the full line will be the average between among three different data sets, and the shadow area is the standard deviation from this average. So when we expose the cell to antibiotics, we treat the cell for 2 hours prior imaging, and the same concentration of antibiotics is kept constant during the acquisition. So we start from the no ciprofloxacin sample, and in this case, this is the curve that describes the SOS signal, and we have very few cells that have induced SOS signal. This is compatible with the fact that we may have a little endogenous DNA damage in our sample. Then we expose the cell to 4 nanogram per ml of ciprofloxacin, and in this case, we can observe the distribution is Y broader. We still have a fraction of cell that has not induced SOS, while we start to have a consistent percentage of the cell in our population that has induced SOS. And this is also compatible with the cell size that we observe, where the cell start to appear as elongated. Then, for the highest concentration that we use, that is 14 nanogram per ml of ciprofloxacin, we have a situation in which the vast majority of the cell will have experienced DNA double-strand breaks and will have started the repair process. And indeed, we observe that the vast majority of the cells in the sample are quite bright and elongated. So in this way, we define 2 different levels of DNA damage, and we then looked at the mobility of RecB in these 3 different situations. To quantify RecB mobility, we computed the D star value. The D star is a measure of RecB mobility and can be computed by using a one-time interval mean square displacement. And we are selecting trajectory that have at least 5 localization. Then from the apparent diffusion coefficient, we also take in account the possible localization error on the particle position. And in this way, we obtain the D star. So for each of our condition, we compute the D star distribution of all the trajectory in our cell population. Let's start from the no ciprofloxacin sample. In this case, the distribution is peaked around the value of D star that are higher than 1 micron square per second. And we have that quite a large number of trajectory have a value of D star bigger than 1. When we expose the cell to 4 nanogram of ciprofloxacin, we start to observe that the peak of the distribution is shifted towards a lower value of D star. This is around 0.5 micron square per second, but still have a large fraction of trajectory that have a D star larger than 1 micron square per second. This distribution shape changed dramatically for the highest concentration of ciprofloxacin. For 14, we have that the peak of the distribution is at very low value of D star, less than 0.5 micron square per second, and we still have a tail of trajectory that has a value of D star bigger than 1. Of course, we want to quantify a bit better this behavior and try to see if we can model our distribution. We started from the highest concentration of cyprofloxacin, and as done for other DNA interacting protein, we look for a model to describe the distribution. And we find a model that describes the distal distribution at the highest concentration of cyprofloxacin as composed by 3 different subpopulations of RecB molecule with a diverse mobility. So the first population here is the red dotted line, and those are what we consider very slow RecB trajectory, and those are correspond to the RecB molecule that are on the DNA that are engaged in the repair process. The second distribution here described by the dotted blue line is consistent with the slow RecB molecule, and those are molecules that having transit interaction with the DNA are the one looking for the double-strand breaks. And then there is a third population of molecule here in magenta that it seems doesn't interact at all with the DNA and is freely fast diffusing, moving inside the cell. We, for consistency and to observe how those the different fraction of trajectory described by each subpopulation change, we apply the same description to the other 2 conditions. And in this way, we can compare how the fraction of molecule of trajectory described by each kind of this star would change for different level of DNA damage. And in particular, today I would like to focus on the population of the very slow RecB trajectory. So in this case, we can observe how the fraction of molecules that are very slow on the DNA is increased with the increasing concentration of ciprofloxacin. And this is show how the fraction of very slow trajectory increase with the level of DNA damage, indicating that more DNA ends are broken, more RecB is engaged in the repair process. So we sort of got an overview of how RecB mobility change when there are different levels of DNA damage. But we were also curious to understand what happens when RecB cannot complete the repair process. So we look at the mutant that has a single point mutation that inactivates the nuclease domain of RecB. And this mutant is called RecB1080, has been characterized in vitro. And what has been observed that it still conserves its helicase activity and is able to recognize and unwind the double-stranded DNA. It also still recognizes the Chi sites, but is not able to load RecA. And this is, of course, open the possibility for other repair pathway to happen, but it's still unclear of how RecBCD would be dissociated from the DNA. So in this case, we start to look at this mutant, just not using any antibiotics, but observing in the non-cyprofloxacin condition. In this condition, we should have still a small amount of endogenous DNA damage. And we look at the distal distribution of RecB1080. And what we observed is the distribution has a quite large peak of very very slow RecB molecule. And this was a bit surprising, even if we especially if we compare with the distribution of the wild type RecB. But those 2 samples, they shouldn't have very different level of endogenous DNA damage. What actually could happen here that we see more higher fraction of very slow molecule. And this could be because actually the missing loading of RecA affect the dissociation process of RecBCD, and then the dissociation happened over longer time scale. So concluding, we look at how RecB mobility change during the DNA repair, and the fact that it can be described as composed by 3 different subpopulations of molecules. In particular, we observe that for increasing concentration of DNA damage, the percentage of RecB that are on the DNA are increasing, compatible with the role of RecB in the DNA repair. And lastly, we look at the situation in which RecB nuclease activity was inactivated, and this could affect the ability of RecBCD to dissociate from the DNA. So I would like to thank you, all of you, for joining us today. The Karui lab where this research has been performed, and all our the other co-author and collaborator, and also our funding agency. And I'm happy to take questions later.
Cool, thank you, Alessia. So we'll move on to our second invited speaker. So if you have any questions about that, do add them into the question box. They'll be asked as a group at the end. Our second speaker today is John Young from the University of Oxford. John joined Carl Robinson's lab as a postdoc in 2020 and uses mass spectrometry and mass photometry to study membrane proteins involved in multi-drug resistance in pathogenic bacteria. John's research is funded by a fellowship from the Canadian Institutes of Health Research, and before joining the Robinson Group, he completed his PhD in membrane protein biochemistry in Vancouver. Today, John will present his work entitled Quantifying Receptor-Ligand Interactions Using Mass Photometry. So I'll just hand over to John, and yeah, this slide looks fine, so good to go.
Hi, everyone. Good afternoon. I'm excited to talk to you today about some of the work I've been doing during my postdoc at the University of Oxford in Carl Robinson's lab in the Department of Chemistry. Before I start, I want to say again, like, all this work was done at the University of Oxford. Our lab, the Robinson Group, is based in the Kavli Institute for Nanoscience Discovery. Research in the Robinson Lab is funded by the Wellcome Trust. I am supported by a postdoctoral fellowship from the Canadian Institutes of Health Research. A big thank you to my collaborators, Professor Philip Kukura and a postdoc in his group, Dr. Emmanuel Fitzner, who really helped me a lot with the mass photometry analysis that I'm going to show. So a constant theme that has run through my research career, both as a PhD student and now as a postdoc, has been the study of membrane proteins, which sit in biological membranes and exist at the interface between the extracellular environment and the intracellular environment. They're responsible for many critical life processes, including nutrient transport, host-pathogen interactions, antibiotic resistance. So they are very important to study so that we can understand these fundamental processes. However, membrane proteins, they sit in the hydrophobic interior of biological membranes, so they can be challenging to study. How do we get membrane proteins out of the lipid bilayer so that we can study them? Often that's done by solubilizing the biological membrane with detergents. The proteins can subsequently be purified and characterized by structural or biophysical methods. Now, detergents can be really successful for characterizing membrane proteins in a lot of applications, but sometimes with certain applications, the presence of detergent micelles can complicate protein analysis. So how do we solve that problem? We can use a so-called membrane mimetic technology, which is able to shield the hydrophobic transmembrane segments of the membrane protein from the aqueous environment. So throughout this talk, I'm using a membrane mimetic called peptidiscs, which is composed of a short amphipathic peptide, which wraps around the transmembrane segments of the target protein. Peptidist reconstitution, it's really quite simple, and it can be readily incorporated during membrane protein affinity purification. So as a postdoc, I'm using a single molecule level technique called mass photometry to characterize proteins in aqueous solution. Mass photometry is a really nice way, really fast, really accurate method of determining sample molecular weights. And it also gives you a really good idea of your sample heterogeneity. How does the technique work? We start with a thin sheet of glass called a cover slip, and we shine a laser beam through it. We apply a small droplet of liquid onto the top of that cover slip containing a nanomolar level of our protein of interest. The molecule of interest lands on the glass cover slip. In so doing, it scatters light. The intensity of that light scattering is directly proportional to its molecular weight. So a smaller entity will scatter an amount of light. A larger entity will scatter correspondingly more light. Those light scattering events are recorded as a movie and are analyzed in the software package to produce these histograms. So here we see species, light scattering from species A, B, and C of different molecular weights. We plot the intensity of the light scattering as a function of molecular weight to derive a standard curve. And so we use this standard curve to determine the molecular weight of our sample of interest. So that's all very well in theory, but how does it actually work? What are the results that we get from this with a membrane protein? So I started with the E. coli, with the bacterial drug efflux pump ACRB. Expressed the protein in bacteria, purified it to homogeneity, and I did a polishing size exclusion chromatography step to ensure the sample is soluble and free of aggregates. Taking a series of successive fractions under the main size exclusion chromatography peak, I analyzed each one by mass spectrometry. And as you can see here from this video, each of these so-called donut shapes represents one distinct landing event. Encouragingly, just by eye, we can see from this video that each light scattering events are roughly the same intensity, which bodes well for the sample and suggests that it is rather homogeneous. So the results from all the 4 fractions, from the successive fractions under that gel filtration peak, are shown here. Reassuringly, they all look about the same. I did this purification procedure twice on 2 independent days. An overlay of the histograms from the 2 biological replicates is shown here, showing that this method is highly reproducible. Good. So, and furthermore, it shows that these samples are really quite homogeneous. The spread of the sample, the heterogeneity in the sample, is less than 5%. That's a good sign. So beyond just measuring sample molecular weight and assessing sample homogeneity, can we use this to study interactions between peptidus reconstituted membrane protein receptors and their soluble protein ligands? As a simple example case, I chose the bacterial SEC-YEG complex, which sits in the inner membrane of gram-negative bacteria and interacts with high affinity with a cytosolic regulatory protein called SID. I purified the SEC-Y complex to homogeneity reconstituted in peptidus. I also separately purified the protein SID. So for the SEC-YEG complex, I chose a single representative fraction under this main size exclusion chromatography peak and analyzed it by mass spectrometry. We derive a mass of about 164 kilodaltons. Addition of a molar excess of the regulatory protein SID to this sample reveals a shift in molecular weight corresponding almost exactly to the size of the protein SID, suggesting that we have a one-to-one binding interaction taking place. Expanding on this initial measurement, I did a binding titration, reproduced multiple times to characterize the binding. We can see the change in peak center mass shift as a function of concentration of SID. We can derive a binding curve and a KD for this interaction, showing tight binding with an affinity of about 200 nanomolar. So that shows that, one, we can use mass spectrometry to see the molecular weight of the membrane protein sample, and we can visualize binding of a high affinity interactor. Moving on to a somewhat more complicated example, I next studied interactions of the bacterial ABC transporter BTUCD with its ligand protein BTUF. Now BTUCD, it's an ABC transporter. It sits in the inner membrane of gram-negative bacteria, and it sits there in the membrane, and it waits for its interactor BTUF. BTUF is a periplasmic protein, so diffuses through the periplasm, scavenging for BTUF, for vitamin B12. When BTUF encounters vitamin B12, it grabs B12 and brings it to BTUCD for transport. So, and this interaction should be at least somewhat dependent on nucleotides, ATP. So let's test that. Let's see if it's true. I purified the BTUCD ABC transporter, reconstituted into peptidus. Analysis by size exclusion chromatography reveals a nice homogeneous peak. I also purified the BTUF protein to homogeneity. Once again, I selected a single representative fraction under the BTUCD peak and analyzed it by mass spectrometry. That produces the black trace here with a molecular weight of about 210 kilodaltons. Addition of molar excess of the BTUF regulatory protein shifts the mass here to the blue curve, which is approximately at 225 kilodaltons. Since the mass of BTUF is 30 kilodaltons, we know right away that we are not getting 100% binding here. So we repeated that measurement in the presence of 1 millimolar ATP, and we get a further shift to about 230 kilodaltons. Still not 100% binding, but that is a significant boost. So I then, moving on from that, I did a full set of binding titrations, producing these K binding curves here. So the information that we can get from this is the apparent affinity, the so-called dissociation constant, is not much changed by the presence of ATP. The KD is still about 50 nanomolar in the presence and absence of ATP. However, consistent with previously published literature, we can see from this very clearly that ATP increases substantially the fraction of BTUF that is binding to the receptor. That is a nice point to know, and it is not evident by other techniques, such as, it's not evident from other techniques used to study protein-protein interactions, such as biolayer interferometry, SPR, and microscale thermophoresis. So that concludes my presentation. I hope that was interesting and understandable by all of you, and I'm happy to take any questions at the end of the session. Thank you.
Okay, thank you, John. As I said, any questions, do put them in the box and they'll be asked as we go down. So thank you, John. So our third and final speaker today is Martina Foglizzo from the University of Leeds. So Martina graduated with a BSc in Biochemistry and an MSc in Molecular Biology and Genetics degree from the University of Pavia. Then she moved to the University of Otago to study the structure and function of ubiquitin E3 ligases and deoxygenating enzymes during her doctoral training. Following completion of her PhD in 2016, Martina stayed at the same institute and the same lab for a postdoctoral position prior to joining the laboratory of Professor Elton Zakiraj at the University of Leeds in January 2019. Martina's research focuses on understanding how ubiquitin signaling and post-translational modifications of chromatin can regulate DNA damage and repair. And today, Martina will present her work entitled The SPATA5-SPATA5L1 ATPase complex directs replissome proteostasis to ensure genome integrity. So I'll hand over to Martina. Yeah, the presentation's there, so good to go.
Great. Thank you very much, Ben, and thanks to the Biochemistry Society for allowing me to present my work today, which is focused, as Ben just said, on the characterization of the SPATA5-SPATA5L1 complex and how this regulates the DNA replication. So just a brief introduction. DNA replication, as we know, is a cellular process that is required for the faithful transfer of genetic information from parents to progeny, and is driven by the assembly and disassembly on and off chromatin of a complex molecular machine known as the replisome, as we can see here. Different DNA polymerases are required for leading and lagging strand DNA synthesis, and these are polymerase epsilon and polymerase delta. Over the past years, different studies have highlighted how disassembling of replisome components that are required for leading DNA strand synthesis is regulated. And so just briefly, this required the recruitment of the kallin ring butyl ligase, kallin-2-LRR1, which assemble lysine 48 linked ubiquitin chains on the MCM7 subunit of the replicative helicase CMG. This CMG is then recognized by the P97 ATPase together with its adapter protein UFD1 and PL4, which triggers CMG unfolding and replissome disassembly. By contrast, how exactly the replisome factors that are required for DNA lagging strand synthesis are disassembled from chromatin is not quite well understood. So to better characterize this aspect, we have been collaborating with Roger Greenberg Group at the University of Pennsylvania, and they have performed tandem affinity purification of POLD3 from HeLa S3 cell lysate, followed by mass spectrometry. The results that we obtained showed a variety of peptides that corresponded to Pol delta subunits as expected, as well as a few peptides that instead corresponding to the ATPases SPATA5 and SPATA5L1. So just a bit of background on this protein. SPATA5 and SPATA5L1 are ATPases that belong to the same family the CD48 family of ATPases, such as the most well-characterized P97, together with NVL, NSF, and the heterohexameric ATPases PEX1 and PEX6. We became interested in this protein not only because of their role in DNA replication, but also because SPATA5 and SPATA5L1 are characterized by different types of mutations or deletions that can be found in patients with neurodevelopmental syndrome and neurological syndromes. These patients are characterized by intellectual disability, microcephaly, seizures, and other type of impairments. So really understanding how this protein function would be really useful and crucial to improve the treatment and life conditions of these patients. So coming back a bit on the structural and functional aspects of this protein, by looking at the protein sequence and predicted dimensional structure, it was really clear that SPATA5 and SPATA5L1 are characterized by a very similar domain architecture as P97, with an N-terminal domain and 2 ATPase region. We know from a variety of studies that P97 performs a cellular function via interacting with different adapter proteins that then allow P97 to selectively recognize substrate and mediate their ATPase-dependent unfolding. We therefore wondered whether SPATA5 and SPATA5L1 also require some adapter proteins to support their function. We therefore performed additional mass spectrometry experiments when we found association of SPATA5 and SPATA5L1, not only with different factor replication machinery, but also with 2 additional proteins, SIMP and C1orf109. What are these proteins? C1orf109 has been a previously uncharacterized protein that is predicted to bind DNA. Interestingly, to us, if we are looking at the cancer deep map, we see that patients who are characterized by C1orf109 mutations show the same phenotypes and symptoms as patients bearing SPATA5 or SPATA5L1 mutations, showing codependency between these 3 genes. SIMP, by contrast, is a CDK2 interacting protein, is important for genome integrity, and also it's been shown to interact with different replisome components. Our collaborators were also able to perform co-IP experiments where we could see direct interaction between SPATA5, SPATA5L1, SIMP, and C1orf109 in HeLa cells, therefore really showing that these 4 proteins form a single entity. Also very interestingly, inhibition of SPATA5, SPATA5L1, or C1orf109 resulting in downregulation of the other 2 protein partners, indicating that the 3 of them are definitely codependent. So in summary, briefly, we have identified a new macromolecular machine which consists of 2 ATPases, SPATA5 and SPATA5L1, together with the interacting partners C1orf109 and SIMP, and we have called this protein 55cc. As I just shown, 55cc play a role in DNA replication, and other studies over the past year have also shown that 55cc is also involved in ribosome biogenesis. The first question we wanted to address are how does 55cc assemble into a molecular machine, whether SPATA5 or SPATA5L1 are forming 2 homohexamers or a single heterohexamer, and if this is the case, which is the stoichiometry of these 2 proteins in the complex, as well as which is the stoichiometry of SIMP and C1orf109 within 55cc. To answer this question, I was able to reconstitute the 55cc complex in a stoichiometric manner after expression in insect cell, and I performed cryo-EM analysis. So this is a bit still ongoing work, but for now we have managed to get a structure of 55cc at about 4 to 5 angstrom resolution, which really shows a structure and a molecular assembly that we will not have predicted, and it is really different from all the ATPases that have been characterized so far. Specifically, the complex is characterized by an upper portion, which we call the lead, which consists of 2 copies each of C1orf109 and SIMP, which are assembled in this diamond-like conformation and that are supported by the N-termini of SPATA5 and SPATA5L1. Quite interestingly and differently from the only other heterohexameric ATPase characterized so far, PEX1 and PEX6, here we have an uneven ratio between SPATA5 and SPATA5L1, with 4 copies of SPATA5 and only 2 of SPATA5L1. And this arrangement, as well as interaction between these proteins, were also confirmed by cross-linking mass spec. By contrast, the bottom part of the complex is characterized by the ATPase units, so SPATA5 and SPATA5L1, and these do arrange in a conformation similar to what has been observed, for example, for P97. So in summary, we've shown that 55cc consists of a 4-2-2-2-2 stoichiometry with 2 copies each of the interacting partners, SIMP and C1 of 109, and for the first time, uneven assembly, so 4-2 ratio between SPATA5 and SPATA5-1 ATPases. The second question we wanted to address is whether or not how 55cc functions as a molecular machine, whether it is an active ATPase, and whether or not it is able to unfold protein substrate on chromatin. To answer these questions, I initially performed EMSA assay testing the ability of 55cc and the isolated SPATA5 protein to bind to different DNA types. As you can see here, both 55cc and SPATA5 were able to interact with all these DNA forms, and the interaction was pretty similar between the 2 assemblies, indicating that SPATA5 does mediate the majority of this interaction. By contrast, C1 of 109 and SIMP did not display any binding. We also moved on and performed some ATPase assay testing and comparing SPATA5 and 55cc in the presence of DNA. So SPATA5 shows some limited ATPase activity that could be, at least in part, enhanced by the presence of DNA. By contrast, 55cc showed a higher ATPase hydrolysis rate, and also this could be enhanced by RNA, by DNA. So these results told us that really the fully assembled 55cc complex is required for proper enzymatic activity. Our collaborators also performed extended Western blot on chromatin fractions, and they were able to visualize these low molecular weight products for a variety of replication factors and components of the replication machinery, such as RFC1, PolA1, PolD3, PolD1, and PolD3, which were not present or strongly reduced when any of the components of the complex were not present. At the moment, we think that replicating chromatin serves as a platform for 55cc to perform its unfolded activity, and when the substrates are unfolded, they are then cleaved in the different low molecular weight fragment that we can observe here. So to conclude, I've shown that 55cc is a new protein complex that is required for processing of replisome substrate. The 3-dimensional structure does show in SPATA5 and SPATA5L1 heterohexamer with C1 of 109 and SIMP bound on the top, forming this substrate entry gate that is required for substrate unfolding. And we also think that 55cc acts as an unfoldase by remodeling protein complexes and facilitating their cleavage or clipping before removal from chromatin. And although I have not had time today, we also have data showing that this process is ubiquitin independent and therefore different from the processes P97 triggers. And we therefore propose that this represents a new mechanism of replication factor proteostasis. And with this, I would like to conclude by thanking Elton and everyone in the lab. A big thanks to our technician, Lisa and Laura. Thank you to the EM facility as well for training and support, and big thanks to Oksana for helping out data processing. Big thanks also to Anton and James for mass spec, and I really would like to thank Roger for allowing me and Elton to work on this project, and a big thanks to Vidya for performing a lot of the cell biology work. And with this, I thank everyone for your attention. I'm happy to take any question.
Okay, thank you, Martina. So we now welcome any questions for any of the speakers. So if you have a question, please type it into the question box. I think I've shown an image on the screen now, and we'll try and make our way through those. So we do have a couple of questions already in, so I'll start with them. So related to genome integrity, so maybe first Alessia. So using your sort of in vivo imaging technique, are there any environmental or different environmental or genetic factors that might influence some of the diffusion of RecBCD? So have you tried different? Well, I guess have you tried different DNA damaging agents or different sort of genetic backgrounds that maybe influence the behavior of RecBCD?
So RecBCD is particularly involved in DNA double-strand breaks. So we had to make DNA double-strand breaks. And we chose ciprofloxacin because we could modulate it and give the possibility to RecB to repair the damage. Indeed, we are using sublethal concentration, and hence actually the cell can repair. And we don't see cell die usually. I mean, it's you can notice when the cells are dying. And different genetic background, maybe if could they expand a bit? Meaning we try with the MG1655, like the classic E. coli strain, and okay, so I'm not sure if they meant something. We try to look at physiological conditions, so condition in which E. coli can experience normally in its life cycle this kind of DNA damage.
Yeah.
If they want to expand a bit of that, yeah.
Yeah, I'll see if they had another question. But on that, so you use sublethal doses. Can you follow? So you can follow single cells, but can you follow subsequent generations if they can still?
So usually, what's happening is that so we image the cell for around in total for 40 minutes. So we don't keep the sample longer on the microscope, and we observe that the cell appear quite elongated. So they have it's a bit challenging for them to divide. And especially at 14, because actually at 4 we can still after a while the cell that keep dividing. So I actually checked if they were still doing cell division, and they were. So they're still alive, yeah.
Okay. Cool. I think we're waiting for John to come back online. So Martina, you sort of showed so you picked an independent way of turning over the replisome. Do you think this is a sort of a system that's partially redundant with P97, or do you think it's for specific types of replication stress, perhaps?
So we think it's a process that differs from P97, and that is, yeah, it's probably, we think it's mainly triggered by stress conditions. Yeah, and because a lot of the complex does trigger chromosome instability and separation of the sister chromatids quite earlier on. So we think it's a system that helps the cells to cope with this condition.
Okay. Is that a specific type of replication stress, or is it sort of general?
Yeah, we have not looked at, yeah, we're not looking to the details yet.
Okay, cool. So, John, there was one quick question. So, have you looked at different sort of environmental factors and the effect on different membrane-bound proteins, such as temperature, pH, and how that would affect things like stoichiometry or?
Well, in the protein-protein interactions I was describing, those have been quite extensively characterized by others. Yeah. There's nothing in the literature to suggest that pH or salt concentration or really any other factors would substantially affect those.
Okay. So alongside that, to use these peptidiscs, do alternative lipid environments affect any of this?
Yeah, so very often receptor-ligand binding interactions are strongly modulated by the presence of specific phospholipids. The one factor of peptidist reconstitution that's quite attractive is we have the target protein embedded in a biological membrane surrounded by its functional lipids and bulk annular lipids. We then solubilize that in detergent, and because the peptidist reconstitution process is quite quick, it's quite streamlined, we keep those functionally important lipids present in the disc. They're not removed. So the lipids are there to help promote binding if needed.
Okay, cool. Okay, so another question for John. So I guess in Carol's lab you do sort of native mass spec and things like that as well. Is mass photometry, how sensitive is mass photometry as far as binding of small molecules and things like that? Is that something you can see yet, or is it more sort of protein-protein sort of interaction?
Binding of the small molecule directly, you cannot see by mass photometry. But if a small molecule has an influence on a binding interaction, then yes, you'll see it. So as an example, with the binding of BTUF onto BTUCD, the example that I showed, I did that binding in the presence and absence of ATP. You don't see binding of ATP to BTUCD. We know that ATP binds, but we don't see it directly. But we see the effect of ATP binding on that interaction with BTUF.
Okay, cool, thank you. Quick question, I guess, from Martina. You showed a lot of this sort of heterohexamer in the 55LC. Is it that sort of stoichiometry, do you think, in cells or in a native environment? Or is that more of a case of when you've reconstituted it from insect cells?
I guess we have not done the comparison, yeah. But I think that's how the complex looks like. Yeah, I don't see how that should be different in cells compared to what I do in vitro. I know I can produce SPATA5 on its own, so a SPATA5 can exist by itself. We know the isomologous SPATA5 has function as well on its own in ribosome biogenesis, so there is probably potential that SPATA5 has also a separate role. How that is regulated compared to 55cc, I'm not sure as yet. But if it concerns the stoichiometry, I don't see why this should differ in cells. Yeah.
That's fine. There's a question about computational tools such as molecular dynamics simulations. I wonder if this is more directed towards John, whether that sort of complements some of your work using mass spectrometry and sort of looking at how membrane-bound proteins might react in different lipid environments, perhaps.
So, sorry, what's the question?
So the full question is how computational tools such as molecular dynamics might be important in understanding some of your work or taking that into sort of different lipid environments, perhaps, I think.
Yeah, I mean, molecular dynamics, it's a very powerful tool. So one could envision if you had a membrane protein that was not well characterized, yes, that would be really important to do, because you could use molecular dynamics to understand how your protein complex of interest behaves in the presence of certain types of phospholipids, and that could be really important information for a transporter. It would be important to do. In the Kavli Institute, where my laboratory is based, we have some of the world's leading experts on molecular dynamics of simulations of membrane proteins. I should talk to them about that, because that's an important point.
Another question for you, John. So in some of your titrations, this is from Aoife, you can see a larger component. So the question is, in some of your titrations, you can see a larger component. Do you think this is relevant to the physiology of the membrane proteins you're looking at?
Sorry, I don't understand. What?
So I think in some of your titrations, you see a larger component, or a large component in the mass photometry. Do you think this is something that's relevant, or can you see different? I guess can you see different subcomplexes? I know you titrated it in to try and force the interaction. If you see different subcomplexes, do you think this is something you can is relevant for the physiology of the protein?
Yeah, it's very relevant. So in the examples I showed, it should be a one-to-one binding event. I think the subpopulations that you're talking about, it might be partially dissociated. So like in the example of BTUCD, there's two membrane integral components of the transporter, and then there's two cytosolic components, which can in theory fall off. So you can see that in mass photometry. But yeah, again, like the thing that's really good about the technique is you can see one, what's the fraction bound here, and also what's the stoichiometry of the binding. So I have some data, I didn't show it in the talk, but there's receptor XYZ in the membrane. We titrate in a ligand that binds on the extracellular side, and we can see by mass photometry that this ligand binds in a one-to-one. There's a one-to-one binding mode and a one-to-two binding mode.
Okay.
So you can see that directly by mass photometry, and it's quite difficult to tease that out with other methods, which is quite a nice advantage.
Okay. Cool. So I had a question for Alessia. You showed some data looking at a particular RecBCD mutation. I guess if you looked at mutations in the other sort of subunits using this output, so it has a different components as a complex. Do they have different behavior in your system?
So we start to, but we start with, I showed this particular mutation that we want that investigate better, because we found that was particularly interesting, try to understand how the dissociation process could be affected by a mutation in the nuclease domain. And this is also a mutation that, so it's clearly not allowing the finalization of the repair. So we built some mutant when we knock out the helicase domain, but those are also mutants that are very sick. So they're a bit more difficult to image and grow, while the RecB nuclease mutant is more sensitive to DNA damage, and we actually checked this by looking at the spot test, but it has some ability to repair it, and the reason I say that there was probably another repair pathway involved, jumping to allow the repair up to a certain point. Of course, the cells are more sensitive in general to DNA damage.
Okay, cool. Thank you. I think we're going to slowly wrap up if there are no other pressing questions. So thank you all for attending, and thank you to the speakers for presenting today. You can continue the conversation online if you wish, so following @BiochemSoc or @PP Publishing on Twitter. I think the Biochemical Society now has an Instagram account as well, so check that out if you're into that. We welcome suggestions for future topics and speakers to feature in this Biochemistry Focus webinar series. And if you do have a webinar or a webinar idea for 2023, we invite you to submit a proposal for that by website. And you can find more information about the webinars, propose a webinar, and watch previous recordings at www.biochemistry.org. Any upcoming webinars are listed on the website as well. And if you've missed any of the over 50+ webinars that we've already run as part of this series, or would like to watch them again, please visit the website or the YouTube channel associated with the Biochemical Society. The recording from today's webinar will also be available to watch within the next 2 or 3 weeks as we get the sort of transcript attached to that. And our next seminar or webinar is on the 30th of May. This is at 2 o'clock. The next webinar is 60 Years of the Colworth Medal, a view across the decades. And this session will bring together past Colworth Medal winners from across the decades to discuss their leading research and achievements, share their advice, and answer any audience questions. So it's a great opportunity to ask questions from previous Colworth Medal winners. Finally, I want to highlight that it's more important than ever to stay connected and engage with your fellow molecular bioscientists. You can join the Biochemical Society's community of researchers and specialists and take advantage of an array of key benefits, and this includes discounted registration fees for society courses and meetings that take place throughout the year, exclusive access to a wide range of grants and bursaries, personal online access to 2 of their journals, and much, much more. So do please visit their website with much more information. And if you want to get more involved, there are always calls for joining committees as early career researchers, whether PhD students or postdocs. And it's a great way to get involved and sort of network and see what goes on behind the scenes. So with that, I would like to thank everyone for being here and the speakers especially, and goodbye and have a good rest of the day.
Thank you.
Thank you.